Good morning. I'm Bert Hakkinen, and Tom McCone and I are the co-chairs of the ISEE-ISES Awards Committee. And the Excellence in Exposure Science Award is being awarded for the first time this year, and it was inspired by the work of visionary individuals who helped shape the science or the field of exposure science, as well as supported the origins and growth of ISES. So the first ever recipient is Dr. PETROS KOUTRAKIS, and I will read what the plaque says, and we will have a photograph right afterwards. But it says Dr. PETROS KOUTRAKIS, Professor of Environmental Sciences, the T.H. Chan School of Public Health, Harvard University. It is in recognition of the production of significant advances in exposure science and for leadership and service. So. I believe so, yeah.
Good morning. It's a great pleasure to be here. I'm very thankful that the committee selected me. Before I start, I would like to thank or acknowledge the many people that they helped me to advance my career over the years. And that's going to be a long list, so I grouped them in my colleagues at Harvard, my research team, our very bright students, and the collaborators I had over the years. I also would like to thank two individuals, Dr. Lance Wallace, who nominated me, and also Professor Jack Spingler, who invited me to join his group 34 years ago. Also, if you believe in destiny, the first study I did in North America was in Canada, near Danville, by the Lake Erie. About 32 years ago, we studied the effects of acid aerosols and ozone on children, respiratory health. And since then, I have built many friendships here in Canada. So I'm very pleased to be here in Canada to receive this award. I'm going to talk about something relatively new, something we have been working over the last 2 to 3 years. Much of the work I will present, it was just published or it's under review. So I will ask you to sign a confidentiality agreement, that one that Mr. Trump asks his collaborators to sign. So I make sure you don't really go out with all this information. The motivation for this work is that we try to understand why particles are so toxic. What are the biological or chemical or physical properties which make them so toxic? And the hypothesis here for our studies is that particles act as vectors, that they carry radionuclides, that they can enter our body after inhalation, and they can cause damage due to their reactivity. And radionuclides are atoms with high nuclear energy that they release it in the form of alpha, beta, gamma radiation. And from these 3 different types of radiation, alpha is the most toxic, because it has a large mass, a lot of energy, and cannot be transported over long distance. It goes very short length and damages the cells which are around it. So this is what we call low energy transfer. And for the purpose of this presentation, I'm going to use the word particle for radiation and PM for ambient particles that you and I know. So I'm not going to go through this, but I want to note that the particles that exist in the ambient air, they are receiving radionuclides from many sources. Like one example, for instance, is the beryllium-7 that comes from outside the solar system, or potassium-40 or uranium-235 that they come from the ground, or the progeny of radon. And we are interested here in inhalation, and we're interested in alpha particles because these are the ones that they can be only enter the human body through inhalation or ingestion. Now, in the absence of artificial isotopes, like the ones that they are released during nuclear weapon tests or through a nuclear power plant accident, in the absence of these artificial isotopes, the most important source of alpha particles that they charge the ambient particles is radon. Radon is a gas that decomposes to progeny, which are ultrafine radioactive particles, which attach to the ambient particles. And we know them because of these progeny are well-known lung cancer risk factors. But very little is known about the non-cancer effects of this progeny. And what I'm going to do today is to convince you that there are many other effects, and these effects are very similar to the ones we find for ambient particles. So for the entire presentation, our hypothesis is that ambient particles carry radionuclides that they release alpha particles, which are the ones that they are toxic. I'm going to review some studies, and the first one will be about trying to understand whether radon in the United States modifies the effect of ambient particles. And there are data, not perfect, that they show radon concentrations by county. And we took, for 108 cities, the effect estimates for PM2.5, and we meta-regressed them or radon and other variables, and indeed we found that radon modifies the effects of PM2.5. And just an example, for total mortality for 10 micrograms per cubic meter, the increase in mortality is 1.95 for the cities where the radon levels are, counties actually, below the 10th percentile, and for the cities or counties where the radon is above the 90th percentile, the effect is 3.69, which is almost twice as much. We did another study where we used the Medicare cohort or data, where we study the effect of radon on mortality and case-specific mortality in 63 counties in the Middle Atlantic and Northeastern United States. For those counties, we had both PM2.5 and radon exposure estimates. This was a 14-year follow-up, included over 40 million person years, and individuals, of course, above 65 years old. And we use a Poisson survival analysis. And here you see the effects after controlling for PM2.5. We did not find interaction between PM2.5 and radon, and these effects are just per IQR of the log of radon. And as you can see, the effects are quite significant, and especially for diabetics and individuals with heart diseases. So if we hypothesize that particle radioactivity is the cause for the effects we find, we should be able to take the particle filters and measure some sort of radioactivity property that we can link to the effects. So it could be gross or total alpha radiation, or gross beta or gross gamma, or we can analyze specific radionuclides like bismuth-210 or polonium-210. And this is a good idea, but the problem is that we don't really have good data sets. Luckily, there is the RadNet, the EPA radiation network, that measures gross beta and some radionuclides for almost 30 years. But the main purpose of this network is to identify terrorist acts or nuclear accidents. And that means that the measurements are multi-day measurements. They wait a week or 2 weeks after they measure where most of the radioactivity is gone. And also there is another network that measures gamma radiation. This was a network which was put together after September 11. Again, the measurements are qualitative. However, we have developed some manipulation statistical methods that we can use to adjust and calibrate these data so we can use them. So today I'm going to present a couple of studies that they rely upon gross beta and gross gamma. And the first I want to say is that if you follow the decay chain of radon, unfortunately I don't have a pointer, but you go from radon to polonium to 18, lead to 14, bismuth to 14, lead to 14, polonium to 14, I'm sorry. And then this happens very fast. And then lead to 10 has a half-life of 22 years. So things slow down. And then when lead decays, it decays to bismuth to 10 and polonium to 10. Polonium to 10 is very toxic. And this happens in the order of hours or days. Just keep that in mind. So if we take a filter and we measure the gross alpha and the gross beta, we find that there is a high correlation between gross beta and gross alpha. So for my studies, because we don't have alpha measurements, we will use the gross beta and the gross gamma as surrogates of alpha radiation. So the first study here tries to investigate the relationship between PM, gross beta activity, and blood pressure in the normative aging study, a study supported by the VA administration, a study of men, veterans, where they have frequent checkups. And we use, as I said, the beta measurements of the total suspended particles, not PM2.5, and link them to blood pressure. We use mixed effects models. And I don't know if you can read everything, is that on the left side, you have systolic blood pressure measurements. On the right, we have the diastolic blood pressure measurements. The red bars, they show the effect of just beta at different time windows, up to 28 days. The blue shows the effect of beta after controlling for PM2.5 or black carbon or particle number. You can see that the beta has strong effects, and even if we control for traditional air pollution exposure metrics, that effect remains. And this is the case for both the diastolic and systolic. You can see that there is a decrease for black carbon, and the reason is that there is a correlation between radon and radioactivity and black carbon, since are both local and they are affected by the same meteorological properties like boundary layer and wind speed. A similar analysis was done for lung function, for forced vital capacity, and for the flow expiratory volume. We have on the left the red bars. They show the effect of beta. You can see that there is a decrease in lung function. And even if we control for PM2.5 or black carbon, the effects remain strong for both the FEV and FVC. We have used PM gamma activity, and we found similar effects for respiratory function. And also we have a paper which is ready to go where we find associations between PM gamma activity and pro-inflammatory and inflammatory markers. And actually we find similar effects for beta activity. So I think we have evidence that particle radioactivity really causes effects very similar to those we find. So in order to study these effects in a more systematic way, using more accurate, more quantitative exposure metrics, we need to develop methods. Unfortunately, there is not much out there. So we took different parts and we put our samplers together. This is a study in Boston we just started. On the left side, we have a microenvironmental sampler that measures radon and particle alpha activity. Remember, so far we use beta and gamma because we don't have methods for alpha, for integrated measurements. So what we did here, we make a chamber. Unfortunately, I cannot show the parts. Inside the chamber, the particle, the PM air sample comes inside, goes down to a filter. During the collection, the filter releases alpha radiation, which goes to the top. At the top, there is an electret filter that, collects the alpha particles, and we measure the voltage before and after, and based on that difference, we measure the alpha radioactivity of the particles. On the right chamber, we have a filtered air, and the reason is that there is some radiation that comes from radon that we need to discount, and at the same time, this method is a passive measurement method for radon. And briefly, we don't have many results, but you can see that in the basement you find more radon as you expect, and also you find more alpha particle radiation as you expect. And we have a correlation between PM 2.5 activity and radon. We are going to collect more data. So remember I showed this decay chain, and from the exposure point of view, we would like to know whether we can have biomarkers of exposure and toxicity. If we claim that these radionuclides are the culprits, we need to have some evidence. So polonium-210, that one that comes after lead-210, is a very toxic compound. And actually, polonium-210 was discovered by Marie Curie. Actually, she died from aplastic anemia. Her daughter and son-in-law died from cancer. Her husband died on a car accident before probably he died from cancer. So when you hold and you handle polonium, it generates gamma radiation that can cause cancer. The alpha radiation that causes, it does not really go through the skin. So the Russians, they came up with an idea. How about if we have somebody eat polonium? And what happens when the polonium gets systemic inside your blood? You have lethal properties, and the cells die slowly, and of course, you have death. So can we find these things in human tissues? Of course, we have preliminary results here from 26 autopsies from Brazil. We have more autopsies that they are coming up. And we found that polonium-210, the one I mentioned to you, it's one of the most toxic substances, was found, of course, in the lungs and was correlated with black carbon, but also was found in the olfactory bulb and epithelium and the frontal lobe. And interestingly, the highest concentration was found in the olfactory bulb. And we know from previous studies that ultrafine particles have been found in the brains because they can come and decoagulate. The coagulations together, decoagulate, separate, and they make it to the brain. So I think this is a very concerning finding, and we also have measurements for lead-210, which are kind of similar. So to wrap up the next three slides. I think we have a lot of work in front of us. I think this is an exciting and emerging field. We need good exposure assessment, good measurements. We have a multiplicity of exposure variables we can use. We can go with radon if we want, where there are data on county, or we can do home measurements. We can use gross radiation from particles. The data we had before come from TSP, total suspended particles. We can now use PM2.5. We can do specific radionuclides. They don't have to be specific to radon. They can be specific to cosmic radiation or to solar radiation or specific mining activities. And we can definitely have biomarkers of exposure and toxicity. And because I have a minute, we have done studies, preliminary studies that they show all these outcomes linked to specific properties. And I think the simple thing that everybody here and us can do is to take existing studies and existing outcomes using the same methods and just use different exposures, radioactivity, and try to see what really happens. So it's not a big investment. I show you many, unfortunately, most of the studies I show you are under review or just published, but I believe in totality, there is strong evidence. This is not just, gee whiz, we got a paper, get excited. This is, we have much more information that really substantiates the importance of radioactivity. Now, I don't know, and we don't know whether radioactivity is responsible for the totality of PM toxicity. We don't know because we don't really have good exposure data, and this is what we need to do in the future. Also, we will have challenges in terms of the exposure, so we need to reinvent or design the existing networks or design new ones. And I just to throw this as a last comment is that the radiation, today we focused on radon and alpha particles, but I think the issue of radiation, whether it's terrestrial or solar or cosmic radiation, is going to be an issue that we are going to have to study in great detail in the future. So I think that completes my presentation. Thank you so much.
Thank you so much for that excellent talk. We actually just have time for one quick question. If anybody wants to be that person, there's a microphone here. If you could come on up and just tell us your name and affiliation and a quick question. Anybody?
Hi, Tim Tuccaro from Simon Fraser University in Vancouver. Thank you for that really interesting data. And I just have actually a report from the People's Autonomous Region of California related to your exposure. Yesterday, the Senate passed AJR 30, which will urge the U.S. Congress to speedily take up and pass Restricting First Use of Nuclear Weapons Act of 2017. This is an important bit of news from California, and I hope it spreads.
Thank you so much.